Hey guys, welcome back to Unstuck Biology. So in today's video, we are going to study about the tools of recombinant DNA technology. So I hope guys you would have gone through the last video. Basically, we have started off with a new chapter, which is biotechnology, principles and processes. And in the last video, we actually discussed what are the principles of biotechnology. And basically, we discussed about some tools which are required to accomplish recombinant DNA technology. And today we are going to study all about those tools. So in the last video, we studied about restriction enzymes, we studied about plasmid, and how do we exactly, combine them to form vectors, and then we clone them, right? So let's start off with the first tool of recombinant DNA technology, which is a restriction enzyme. So, guys, what exactly are restriction enzymes? These are the enzymes which basically cut the DNA that we want to integrate into the plasmids. If you remember the last video, we studied that these are the enzymes which are responsible for cutting of DNA. And that is why we studied about a word which was molecular scissors. So basically, they are known as molecular scissors. Why? Because they cut the DNA, right? So basically, guys, in the year 1963, we found out that there are two enzymes which were responsible for restricting the growth of bacteriophage, right? And these were isolated, right? And these were introduced in E. coli. Now, what happened after they were introduced is one of the enzyme added methyl group to DNA, right? And the other enzyme basically cut the DNA. So the enzyme which actually cut the DNA was then known as restriction endonuclease. So this was the enzyme which was restriction enzyme and it was known as restriction endonuclease. Now the first restriction endonuclease which was isolated and was characterized was HIND2, right? So it was HIND2, right? Now what happened is that this enzyme always cut DNA molecule at a particular point. And how does it recognizes that point? So basically, this enzyme recognizes a specific sequence of six base pairs, right? So this enzyme recognizes the sequence of six base pairs and it joins and cut the DNA. Now, this specific base sequence is known as, so these basically base sequences which the enzymes recognize are known as recognition sequence. These are known as recognition sequence, right? So basically, now this six base pair sequence for HIND2 is the recognition sequence, right? Now, if we talk about today, we have more than 900 restriction enzymes, and these are isolated from around 230 strains of bacteria. 230 strains of bacteria, and they recognize different sequences, right? Now, how do we name these enzymes? Why is it called as HIND2, right? So basically, the name of the enzyme has, various letters and numbers, right? Now, the first letter is always of the genus. So basically, we are talking about genus of the bacteria from which it is isolated. And the second two letters, the second and third letter, is species of that bacteria. So basically, if we talk about, say, enzyme eco R1, this is eco R1, right? So in this, the first letter and the second and third letter, right? This is from genus and species name of bacteria, which is E. coli. So E and co. This is eco and RY13. So this is basically the bacteria, right? So it comes from E. coli RY13. Now R is derived from the name of the strain. So the strain of bacteria gives the word letter R. And then we have Roman numbers. Like here we have 1. In case of HIND2, it is 2. So this Roman number is nothing but it is indicating the order in which the enzymes were isolated from that particular strain of bacteria, right? So in the order like R1. So this is the first restriction enzyme which was isolated from the strain EcoR Y13, right? So this is the first enzyme, and that is why we have Roman in Roman numbers, 1 associated with it. Right. Now, the other thing is, guys, that restriction enzymes, these are basically enzymes, right? And there are various classes of enzymes. You must be knowing that, right? So basically, this restriction enzyme belongs to a very large class of enzymes, and this class is known as nucleases. So if you get a question that restriction enzymes belong to which class of enzyme? It is nucleases. Now, nucleases can be of two types, right? It can be exonuclease. So they can be exonucleases or endonucleases, right? Now, exonucleases remove nucleotides from the ends of the DNA. Remember always, exonucleases remove nucleotides from the ends of the DNA, and endonucleases just make cuts at specific position within the DNA, right? So what I said, they remove nucleotides from ends of the DNA. And what do they do? So they make cuts at specific positions within the DNA, right? Now, how do these restriction endonucleases work? So basically, guys, each restriction endonuclease function by inspecting the length of the DNA sequence. Once they find the specific recognition sequence in the length, they bind to the DNA and they cut each of the two strands. So we know that double DNA is double-stranded helix, right? So from the helix, they cut basically both these strands from the sugar phosphate backbone. Yeah, right. So you basically attach to the specific DNA recognition sequence, and after attaching, they cut both the strands at the sugar phosphate backbone. And then each restriction endonucleases recognize the sequence, right? How do they recognize the sequence? They recognize the sequence from the palindrome nucleotide sequence. So basically, they recognize a specific sequence, and this sequence is known as palindrome nucleotide sequence. Now, guys, I hope you know what is palindrome. If not, I'll tell you. I'm writing a word here. Just read it out. Malayalam. The word is Malayalam. Now, if you read it from this side, it is Malayalam. Now, just try and read it from backwards. It is again Malayalam. So say any word, right, which you read either from forward side or backward side. If it comes out to be the same word, this word is known as palindrome, right? Now, in the similar way, a palindrome nucleotide sequence is something that you get the same sequence when you read the two strands. So if you read the 5 is to 3, you read both the strands in 5 is to 3 direction, you get the same sequence. And if you read it backwards from 3 is to 5 direction, you again get the same sequence. So basically, I'll give you another example. So say now, there is a sequence GAATTC, right? This is 5 is to 3, right? Now, since DNA is double stranded, we'll have a complementary strand as well, right? So, this is the complementary strand, right? So, basically, if you are going to read it from 5 is to 3, it is GAATTC, right? Now, if you read from the 3 is to 5 direction, right? It will again be GAATTC, right? Here, remember that you have to take into consideration both these strands. So basically, we are not talking about reading the same strand. We are talking about reading the complementary strand, right? So if I'm reading it from 5 to 3, it is GAATTC. If I'm reading it from 3 to 5, it is GAATTC. Now, since the strand sequence is same whether we read it forward or backward. This is known as palindromic nucleotide sequence, right? So basically, these are the sequence which are present in the DNA, and on the basis of these sequence, basically the restriction enzymes, they recognize this specific sequence and they join and they cut the DNA. Right? Now, basically, restriction enzymes cut the strand of DNA a bit away from the center of the palindromic sites, right? And basically, they make a gap in between two bases of the on the opposite strands, and this leaves, a single stranded portion at the end. So there are basically overhanging stretches at one end. And these stretches are known as sticky ends. These stretches are known as sticky ends, right? And why they are known as sticky ends? It is because if they get their complementary cut counterparts, if they get their complementary counterparts, they'll form hydrogen bonds and they will join, right? So basically, this stickiness of the ends, this helps the enzyme DNA ligase, which joins the two strands of the vector DNA and the foreign DNA, right? So this stickiness helps in joining for the enzyme DNA ligase. Now, guys, remember that these restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources or different genomes. So, let's see how these restriction endonucleases actually work, right? Let's see the same thing with the help of a diagram. Okay, this basically is how the restriction enzyme is going to work. Now say this is the vector DNA and this is the foreign DNA, right? Now the enzyme EcoR1, the enzyme EcoR1 cuts both DNA at the same site. So basically this cuts the DNA between bases G and A, right? And this cuts only, always remember that since it recognizes a specific site, a specific sequence, it will cut only when the sequence GAATTC is present in the DNA. So it will recognize this particular sequence and it will cut both the DNA pieces from the same places, right? So we have echo R1, which will join and cut both the pieces. Now what we get here, right, is now hanging pieces, the sticky ends. So we have sticky end here, we have sticky end here, right? Now the DNA fragments will join from the sticky end. So DNA fragment, they join at sticky ends, right? And now what we get here is the recombinant DNA. What we get here is the recombinant DNA. Now let's see what happens after this. What is the entire process of recombinant DNA technology, right? So basically, we'll see it from another diagram. So now from this particular diagram, we are going to understand how recombinant DNA technology is going to happen. Again, now let's see. So basically, guys, this is the foreign DNA, right? And this is the vector DNA, which is basically the plasmid, the circular extrachromosomal plasmid. So what is happening is that the same restriction enzyme is cutting both foreign DNA and vector DNA at specific point, right? So same restriction enzyme cutting both foreign DNA and vector DNA at specific point, right? So once they cut it, you get a piece like this here of foreign DNA, and here the same piece is missing from the plasmid. Now we have what we have DNA ligases, which joins the two DNAs together. So ligases join foreign DNA toward to the plasmid, right? And here, if you notice, guys, this is the sequence of foreign DNA which is combined with the vector now. So this molecule is nothing but the vector. This is the recombinant DNA molecule. Now, once we get the recombinant DNA molecule, we basically introduce it in the bacteria again, and now through transformation, this will get integrated into the genome of that bacteria, right? So here the bacteria is E. coli. Now, as soon as they start dividing, so basically E. coli is the cloning host. I hope you would have watched the last video. We also understood cloning. So basically now E. coli is the cloning host, and as soon as they start dividing, the genetic information is passed to the next generation, and you can see the phenotypic change. So here what is happening is the cells start dividing. The cells divide, and along with that, the genetic material which has been put into are also being replicated, and the phenotypic effects are being shown. So guys, this is all about the recombinant DNA technology. I hope you understood how it happens exactly. We'll just see one more topic, that how do we actually separate and isolate the DNA fragments. Till now, we just understood that after isolating, we integrated, how we cut it. But how do we actually, separate and isolate the DNA fragments? We'll see that as well. So what is happening basically is that we cut the DNA by restriction endonucleases, right? That results in fragments of DNA. Now what we do is that we take these fragments and we separated by gel electrophoresis, right? So since what happens in gel electrophoresis, basically, since the DNA fragments are negatively charged molecules, what we do is we basically separate them by forcing them to move towards anode, right? Now we basically put them under an electric field through a medium. And usually this medium or matrix is a gel based, which is no, most probably which would always be agarose. It is agarose gel. Right. So basically, agarose gel is extracted from seaweeds. It is a natural polymer which is extracted from the seaweed, right? So we put them in agarose gel, and then we use electric field, and they start moving towards the anode. Now, how they separate is according to their size, right? So basically, through sieving effect, they start separating according to their size. Now, what happens is the larger the size, the closer they will be, right? And the smaller the size, the farther they move. So basically, if you see the diagram here, right, what is happening here is that you have the wells, right? You put the DNAs, and then you can see these DNA bands. So if you see the DNA bands, the closer ones are the largest ones, and the farther ones are the smallest ones. So basically, they are moving as per their size. They are separating as per their size. Now, what happens after that? So basically, guys, these separated DNA fragments, these fragments can be visualized only after we stain the DNA with ethidium bromide. We stain them with ethidium bromide. And then what we do is after staining it with ethidium bromide, we expose them to UV radiation. Now, because of this exposure to UV light, you will see bright orange colored bands of DNA. And these separated bands of DNA are then cut out from the agarose gel, basically, and they are extracted from the gel piece, which is known as elution. So this step is known as elution. And then this DNA fragment, they are purified and they are used for constructing recombinant DNA by joining them with cloning vectors. So this is the way through which we separate and isolate the DNA fragments, and then we use these DNA fragments for recombinant DNA technology. We integrate them with the cloning vectors. So, guys, with this, we have finished the first tool of recombinant DNA technology, which is restriction enzymes. So, I hope you would have understood this topic well. Please do not forget to like the video if you did, and yes, do share it with your friends as much as possible. And guys, if you have any doubt in regards to this topic, please do not hesitate in sending me your doubts over the comments. You can also share your doubts on WhatsApp. I have given the number in description of this video. And yes, guys, if you're new to the channel, please do subscribe to the channel and press the bell icon so that you can get all the updates that I put on my video. Thanks for watching this video, guys. See you in the next one. Take care. Bye bye.